La majorité des organismes qui voyagent sur les bouées et les autres supports ne sont pas visibles à l'œil nu. Ce sont ces micro-organismes qui sont à la base de la chaîne alimentaire. Ils colonisent chaque fragment de plastique par milliards. Un chercheur de Sheffield, au Royaume-Uni, étudie ces microbes. 1 gramme d'eau de mer ou de sédiments contient des milliers de micro-organismes très différents. Mais 2 espèces sont majoritaires sur les fragments en plastique. Peut-on parler alors de microbes du plastique? Jesse Harrison a prélevé des échantillons de sable sur plusieurs plages anglaises. Pour reproduire ce qui est devenu la norme au Royaume-Uni, il a mélangé le sable avec du plastique. Des millions de microbes ont ainsi été mis en présence de polyéthylène, la matière plastique la plus répandue au monde. À l'aide d'un microscope à haute résolution, Jesse Harrison a alors observé l'apparition de petits trous dans le plastique. Manifestement, les microbes l'ont grignoté. Le plastique pourrait donc servir de garde-manger aux microbes. Les micro-organismes qui peuplent les océans ne chôment pas. Chacun effectue une tâche et certains attaquent le plastique selon un mécanisme que l'on connaît encore mal. Ces microbes représentent une vraie source d'espoir.
Plastics have very complex structures and are difficult to biodegrade. Anyhow, we were curious and hopeful and still wanted to give it a go.
With this idea in mind, Jeannie and I read through some hundreds of scientific articles on the internet, and we drafted a research proposal in the beginning of our grade 12 year. We aim to find bacteria from our local Fraser River that can degrade a harmful plasticizer called phthalates.
We read that each year, the Vancouver municipal government monitors phthalate concentration levels in rivers to assess their safety. So we figure, if there are places along our Fraser River that's contaminated with phthalates, and if there are bacteria that are able to live in these areas, then perhaps these bacteria could have evolved to break down phthalates.
So we presented this good idea to Dr. Lindsay Eltis at the University of British Columbia, and surprisingly, he actually took us into his lab and asked his graduate students, Adam and James, to help us. Little do we know at that time that a trip to the dump and some research on the internet and plucking up the courage to act upon inspiration would take us on a life-changing journey of accidents and discoveries.
The first step in our project was to collect soil samples from 3 different sites along the Fraser River. Out of thousands of bacteria, we wanted to find ones that could break down phthalates. So we enriched our cultures with phthalate as the only carbon source. This implied that if anything grew in our cultures, then they must be able to live off of phthalates.
Australia and throughout the world, there are millions of contaminated sites. These include old service stations, sheep dips, tanneries, landfill sites, any contaminated factories and so forth. They're everywhere around the world and they have left loads of contaminated soil and water that need to be cleaned up. So how do we do that? How do we clean this stuff up? One way is bioremediation, and this involves using fungi, bacteria, plants or their enzymes to degrade or munch up all those toxic chemicals and turn them into something less toxic or even harmless. In this experiment, we're trying to find some bacteria that can do the job. Now, you don't need to go and sneak into sealed off areas on contaminated sites. You can get this anywhere. I got my soil from a service station down the road from where I live. It's on a little patch of lawn next to the petrol bowser, okay? But you can get soil from anywhere. It could be your compost heap, it could be your garden, it could be, like I said, petrol stations, or if you're on a farm near the chemical storage shed. The really cool thing about this experiment is that it's real science. I have no idea what bacteria might be present in this soil, but what bacteria are there, there's likely to be species that are unknown to science. Will they break down the engine oil that I got from home? I have no idea, but we're about to find out. This is a handy way of setting up your experiment and controls by writing the ingredients for each of the 4 beakers down on a bit of paper. Now, we've got our beakers set up, we've got to put some oil in. 2 mils approximately into each of these beakers. These three are controls. Beaker 4 is the one where we put in everything. And now the nutrients, food for the bacteria. And all 4 into beaker 4. Now for the soil with our bacteria. Soil into beaker 4, soil into beaker 3. So in beaker 4 we have our oil, our water, our nutrients, the food for the bacteria, and our soil. Hopefully with lots of cool bacteria that can munch up that oil. Beaker 3 has just the oil, the water and the soil, but no nutrients. Beaker 2 has just the oil, the water and nutrients, no soil. Beaker 1, just the oil and the water. Okay, so we've got all our beakers, we've got our bacteria hopefully in our soil, but now we've got to keep the bacteria alive. We've given them nutrients, which is food for bacteria, but they also need oxygen. This is what this is for. This is an aquarium pump. It will bubble oxygen into each of these beakers and hopefully keep the bacteria alive. So you've got to put a little bit of foil, stops your water evaporating over each of these beakers. You can use a Petri dish lid if you like. Now, put that in there, in each one of them. Turn her on. And away we go. Once that's started running for every couple of weeks or every few days, as often as you like, over about 2 or 3 week period, come back and do the grease spot test. And we'll show you how to do that next. Okay, this is the grease spot test. This is where we figure out if the bacteria in our soil are any good at munching up the engine oil that we put into our beakers here. We take a sample from each of these 4 beakers. So we take a couple of drops from just under the surface from each of these beakers and we put them onto a piece of brown paper. When those drops dry, they will leave a little grease spot and we can measure the diameter of that grease spot. Now every few days we do the same thing, we take another grease spot test and we measure the diameter of that spot. If that spot is shrinking, that is a pretty good indication that there is some bacteria in our soil that are able to degrade the engine oil. And that's a very cool thing.
Microbes with a natural appetite for oil live everywhere in the environment. They're nature's recycling units, capable of eating toxic oil and digesting it into byproducts, which are then safely consumed by marine life. Researchers have collected microbes from all over the world and mixed them together in combinations which will address nearly all toxins in oil pollution. They're grown in a catalytic solution to reproduce quickly until trillions are available to be packaged, stored in a powder form, and applied when needed to help clean up sensitive environmental areas. Microbe research attracted the attention of Texas Land Commissioner Gary Morrill, who for years had been the state's strongest proponent for clean beaches. The Texas General Land Office funded a test of the microbes in the fall of 1989 to learn more about them.
The test is designed to show the efficiency of using a biological process to remove oil from the environment, especially oil that's on the surface of the sea. It's a process that's been going on in nature for many, many years, and we have concentrated on increasing the activity of the biological process.
First, oil was released into a controlled pond. Next, microbes in a powder form were scattered over the top of the oil with an ordinary flour sifter. Within minutes, the oil slick began to disappear as the microbes did their work, releasing byproducts which are safely edible by marine life. Shrimp, which had been placed in the control tank, began to rise to the top and nibble on the microbe byproduct as it slowly sank to the bottom. Once the oil had been consumed, the microbes began to die off due to lack of a food supply.
Well, most of the hydrocarbon will be gone. If there's any residual, we'll clean it up with paper towels. We don't expect to use many paper towels.
The microbe level in the ponds quickly returned to normal. Water samples were taken for analysis by Lower Colorado River Authority experts and later reported to be non-toxic and effective. Then the megaborg, loaded with 38 million gallons of crude oil, caught fire. For several days it burned out of control in the open Gulf of Mexico. Commissioner Morrow and the Land Office pushed for and finally won the right to use this potential disaster for the first open-sea testing of the oil-eating microbes. The powdered microbes are mixed with water, then applied using standard firefighting equipment. Cost is about 1/10 of the price of current spill response techniques. Again, samples were taken and results measured. Later in Houston, Commissioner Morrow and Texas Water Commissioner Buck Wynn announced the results.
If you can believe your eyes, we had a great success. And if you can believe the scientific results of our small-scale application test, we're on the verge of a major breakthrough. We have absolutely no evidence that there are any harmful side effects from these microbes. It's time for us involved in oil spill response to put bioremediation at the center of our oil spill contingency plans.
And when you look at the tools we presently have to work with, booms, skimmers, dispersants, and absorbents, bioremediation stands out above all of them. Because these naturally occurring microorganisms appear to degrade significant quantities of oil in a startlingly short period of time, and because we have yet to identify any harmful side effects from the products used, I am persuaded that this technology can be developed and used as a valuable weapon in our arsenal to deal with future oil spills.
Little did the two officials know that in about 2 weeks they'd have to make good on their promise. A barge filled with heavy crude was struck by a ship and began leaking its contents into Galveston Bay. This time it was not the open beaches that were threatened, but the sensitive marshes and breeding grounds of many aquatic birds and other marine life. As oil flowed into the marshy areas, cleanup was begun, but workers quickly found standard skimmers, booms, and sorbent materials hardly worked at all. And just walking through the wetlands can harm the grasses almost as much as the toxic oil. Dispersants could not be used because they also were toxic to both wildlife and plants. Application of microbes was again authorized, this time in marked zones, so specific conclusions could be drawn about their effectiveness. Microbes were sprayed with hoses from boats offshore, minimizing trampling of the marsh grasses. Just 6 weeks later, the results were evident. In areas where bioremediation was used, grasses flourished, and the mud banks had returned to normal. Small living creatures returned to the area. All signs of shoreline discoloration had disappeared. In those areas left untreated, marsh grass was brown and dead, and wildlife absent or endangered.
Science. Les chercheurs japonais ont découvert que des microbes pouvaient aider à faire disparaître le césium radioactif de l'eau et des sols. Une découverte surveillée de près, évidemment, après l'accident nucléaire de Fukushima du 11 mars dernier et des relevés de césium préoccupants dans des zones situées entre 30 et 50 km de la centrale. Une équipe du professeur Ken Sasaki de l'université Kodosai Gakuin d'Hiroshima travaille depuis 10 ans sur l'impact de microbes de bactéries phototrophes sur les métaux. Une telle action est possible parce que les ions négatifs sur les microbes attirent les métaux à charge positive. Les chercheurs ont récemment fait l'expérience avec 2,5 grammes de césium mélangé à de l'eau et 90 grammes de bactéries. En 24 heures, le taux de césium est tombé à 1/12 de sa densité originelle. 3 jours plus tard, il avait disparu. J'aimerais pouvoir décontaminer la terre et l'eau irriguée de Fukushima en utilisant notre technologie. J'aimerais expérimenter notre technologie à Fukushima. De fait, pour l'équipe de chercheurs, les microbes pourraient tout à fait enlever le césium radioactif qui est répandu tout autour de la centrale. Elle envisage de faire des tests dans l'eau et dans les sols de la préfecture de Fukushima pour mettre en pratique sa méthode.
The mycelium absorbs the oil. The mycelium is producing enzymes, peroxidases, that break carbon-hydrogen bonds. These are the same bonds that hold hydrocarbons together. So the mycelium becomes saturated with the oil, and then when we return 6 weeks later, all the tarps were removed, all the other piles were dead, dark, and stinky. We came back to our pile, it was covered with hundreds of pounds of oyster mushrooms, and the color changed to a light form. The enzymes remanufactured the hydrocarbons into carbohydrates, fungal sugars. Some of these mushrooms are very happy mushrooms. They're very large, showing how much nutrition that they could obtain. But something else happened, which was an epiphany in my life. They sporulated, the spores attract insects, the insects laid eggs, eggs became larvae, birds then came bringing in seeds, and our pile became an oasis of life. Whereas the other three piles were dead, dark, and stinky, and the PAHs, the aromatic hydrocarbons, went from 10,000 parts per million to less than 200 in 8 weeks.